Thank you for giving me the opportunity to give you an update on a story which began a long time ago, actually. Haemophilia was seen as early as the late 1970s as a very suitable target for treatment by means of gene transfer for a variety of reasons, which I'll just list next. We started a successful trial here in London at the Royal Free in 2010. So I've got 10 years of progress and problems to talk about. I do have financial interests, which are mentioned here. Why haemophilia? Well, monogenic, albeit there are 7 or 8 different types, depending on which clotting factor in the coagulation cascade is missing. The 2 commoner ones are the X-linked haemophilia A and B, factor 8 and factor 9 deficiency. A wide range of level of those factors can more or less correct the bleeding tendency, which is the entire problem in haemophilia. If you lack those factors, you can't make thrombin quickly enough in the right place to stop bleeding from any vascular injury. You don't have to make the protein in any particular tissue, unlike in many other disorders that have been mentioned, provided it gets into the bloodstream. That's where all the other factors are, and you can reconstitute a normal thrombin generation cascade. We have animal models that we've had for 50, 60 years, and the ideal would be a one-time treatment. Current treatment involves repeated infusions 2 or 3 times a week, although somewhat less often with recent advances. The current therapy is certainly expensive. It gives rise to a $10 billion annual turnover for making replacement clotting factors. And if we could get this right, it could be a global treatment for all patients, not just those in the wealthy countries where we can afford and have the infrastructure for replacement therapy. This tells you pretty much all you need to know about haemophilia, really. These points on this graph represent the annualized bleeding frequency into joints, even on good replacement therapy, in individuals with different residual levels of clotting factor 8 in this case, ranging from 0 in the most severely affected up to the more mildly affected patients who might have 20, 30, or 40%, depending on the different mutation. And I said this is monogenic, but it's actually highly diverse at the level of the mutations. We set up a database some years ago which has more than 10,000 different mutations causing haemophilia A. So unlike sickle cell disease, where you might be attractive to target a specific mutation, that's not really practical, at least with current technology. I just say that in passing. But the traditional division into severe, less than 1%, moderate, less than 5%, and mild, about 5%, was borne out by this study of individuals with different levels of residual factor. What should we aim for? When we started out, 5% looked like a pretty good target because it would drastically reduce bleeding. Subsequently, we thought about going up over 15% because then you don't get any joint bleeds, and the long-term effects of joint bleeding are the cause of the main physical burden in haemophilia, as you'll see, not in this country, but if you go to a third world country and go to a haemophilia clinic, most of the patients are young. The life expectancy is under 20 for severe haemophilia without treatment, and they'll all have severely, increasingly damaged, crippling haemophilic arthropathy due to bleeding into the joint space, which rapidly destroys cartilage. How high should we aim has been a matter of discussion in the haemophilia clinic after our first studies, and several of the authors of this paper, by the way, are people who themselves have haemophilia. Others are haemophilia treaters, and the conclusion from interviewing many people with haemophilia was that the ideal would be to get up to normal hemostasis optimal health and well-being. Actually, anything, as I've already mentioned, could be good, minimal joint impairment at this point, but you still have the problem that an injury or surgery might mean, certainly would mean, if you're undergoing brain surgery, for example, that you need completely normal hemostasis. And by the way, now that we have pretty good haemophilia replacement therapy in this country, life expectancy has risen almost to normal, but what keeps it off normal is now the commonest cause of death in older haemophiliacs, and many, many are reaching older ages. Now that the catastrophe, of course, of HIV, AIDS and hepatitis C has passed through, and we have effective treatment for both of those, the residual elevated death rate comes in middle and older age from cerebral intracranial hemorrhage. We are using a favoured vector, modified virus, of the not a coronavirus, although it looks a bit like it. It actually travels around with a coronavirus, the common cold, which most of you in this room are probably suffering from at the moment. The adeno-associated virus is a dependovirus, contains a single-stranded DNA, and it requires to co-infect with an adenovirus to go through its replicative life cycle. The wild-type genome of the virus is about 4 kb. It's a single-stranded piece of DNA, has the rep and cap genes that are responsible for making the capsid, which is an icosahedron. It has 20 subunits, so you only have to code for one of them, and then it self-assembles. That's a trick that several virus families have invented in the course of evolution. The maximum packaging size for the DNA into this capsid is around 4.5 or possibly 5 kilodaltons, which is a bit restricting when you come to factor 8. For factor 9, which has a small DNA, the protein coding sequence is less than a kilobase. It's not specifically a problem. Here is one expression cassette that we're currently using in our freeline study. There are others available as well, in fact, many others, as we'll see. Just in passing, I should mention there are many serotypes of adeno-associated virus which have been isolated from human or from primates, or in one case from goat. And these have different tropisms, including to heart, skeletal muscle, liver, AAV8, AAV5 as well to liver, and AAV6 to central neurons. So there's many opportunities to use this vector system for gene transfer into various tissues. The one tissue we've chosen to target in the studies I shall describe is the liver. It's where all of the clotting factors are made, factor 9, except for one, factor 8, which it turned out quite recently was eventually proved to be made in blood vessel lining cells, endothelial cells, along with its carrier protein, von Willebrand factor. So targeting the liver to make factor 8 is actually a heterologous tissue, the hepatocyte, but as I mentioned earlier, it doesn't matter so much where you make the protein as long as it gets exported to the blood. The AAV transduction pathway is sketched here. I won't spend much time on that. This time is short. Suffice to say that the particular property of the AAV that's so attractive to us gene therapists is that it will transport your DNA of interest into the nucleoplasm, where as a single strand it needs to be a second strand synthesis. That is a point at which some single strand DNA nucleases can go to work on it. You can make a self-complementary vector. One of the studies, the initial study we did, used the self-complementary palindromic sequence, which automatically self-pairs, improving efficacy. It then forms into concatenars, which are not mostly integrated into host chromosomal DNA. They exist episomally. And that gives rise to the issue that if you have a rapidly dividing target tissue, those could be lost. There are some barriers, immune barriers to this. pre-existing antibody. All of us get infected with AAV2 in childhood and possibly other serotypes. So if there is a presence of significant amount of neutralizing antibody in patients' sera, then they're not going to respond to this mode of gene transfer, and we therefore test for this. And in most populations, between a third and a half of the people with haemophilia that we test turn out to have neutralizing antibody. We're working on ways around that, but for the moment it's a barrier to entering a study. Now, transaminitis is something that we've observed in all the studies so far where the dose level has been increased above a certain point. And one concept for how this happens could be that vector capsid peptides are displayed in the context of MHC class 1, giving rise to a T-cell mediated attack, liver cell damage, and loss of expression. And we do have evidence that that is part of the story in some of the studies. Now, two key studies I'll just briefly present here. The first one was from a group sponsored by Avogen, including first author was Mano and Kathy High's experimental work at the Children's Hospital of Philadelphia was key to building up towards this. They used an AAV2 vector with a factor 9 expression cassette, a dose of 2 times 10 to the 12 vector genomes per kilogram, their highest dose in this study, led to elevation of factor 9 level in a subject with severe haemophilia B, which reached 12%, but then within a couple of weeks started to fall at the same time that the transaminase, alanine amino transaminase, rose as the factor 9 level fell. And it was at this point that it was found that T-cell specific to vector capsids were present, and that was concluded to be why there was a loss of transvected cells and a loss of expression. We were able to use this study as a launching point to design a second trial in which a self-complementary expression cassette was used contained in AAV8. Because AAV2 isn't very hepatotropic, the vectors were infused directly into the hepatic artery in this study, whereas because of the hepatotropism of AAV8, we were able to give this through peripheral blood infusion over the course of an hour at a dose of, in this case, 2 times 10 to the 12 vector genomes per kilogram, same as in the earlier study. And here we notice that someone, a participant who at this point has a factor 9 level because of a previous infusion, part of his prophylactic regimen, that falls but never falls below about 6. At this point, week 8, his ALTs rose. We were ready with a short course of prednisolone orally, which did rapidly cause the ALT to fall. However, he had lost a bit of expression. Subsequently, nearly 10 years later now, his level's still running at 2%. The next patient, we were kind of alerted to this. We thought we'd got away with it because we'd gone beyond the 4-week time point there in that patient. But this patient had already been dosed, and so we were watching with very, very frequent ALT monitoring. It started to go up here. We came in quickly as the factor 9 level fell with a prednisolone. 9 years later, his factor 9 is actually still running along over 5%, and he has not had any spontaneous bleeding in that time or any administration of factor 9. We published that at the end of 2011, and then a follow-up in 2014 with 6 patients at that point treated at our high dose level, all of whom attained a clinically significant level of factor 9. And some of them had a transaminase response that we treated with oral steroid. 2 of them didn't. They've all continued to have the same level of factor 9 expression up to 10 years later that they had after 12 weeks after infusion. Now, factor 9 usage was reduced to near 0 in 6 of these 10 patients, the 6 treated at higher dose level. We've seen no lasting adverse effects. This publication certainly caught the attention of industry, disruptive therapy, obviously, and since that time, I think somewhere in the region of $7 or $8 billion have gone investment into gene therapy. These studies the study that I mentioned here was supported by NIH and Wellcome and MRC. So from small beginnings, great results can emerge. Now, 5% evidently and 6% is enough to have a good result in many patients, but it certainly doesn't restore normality. How could we get to higher factor IX levels? One happy experiment of nature is the mutation that was first noted in a family in Padua, where a young man presented with a very severe iliofemoral vein thrombosis, and the only abnormality found was that his factor IX level was 800%. And the factor IX protein level was normal, 5 micrograms per mil, but it had an enhanced catalytic activity due to a single amino acid substitution, arginine leucine, at 348. I think I've remembered that right. And so by introducing that mutation, you can get a big increase in your activity for a given amount of protein expression. 2 studies are reporting, and I'll mention ours, a 3rd study. And in this study, SPARC study, which is Kathy Hy's company, who were recently bought by Roche for $4 billion, I mentioned. They have attained a mean factor IX level in the 30s, which is good enough to stop all bleeding. Unicure, using the exact construct that I described earlier in our original study, have got similar results. That technology was licensed to them. Another way to improve expression would be to increase efficiency of vector transduction. These use rather standard AAV vectors, and another approach is to anticipate and control the transaminitis. Just a publication here from the SPARC study with some more data showing individual patient responses. There's quite a wide variation here. We don't understand that in full, but it is a feature of all these gene transfer studies in haemophilia to date, so that patients can be offered a clinically meaningful result, but the exact level reached can't currently be guaranteed. A couple of the studies in that, patients in that study did have transaminitis, the others didn't, and this was treated with prednisolone, in one case with loss of activity, and the other without loss of activity. Again, not well understood at this point. We can't do repeated liver biopsies in our patients with haemophilia to get a closer look, although we have now got ethical permission to do it in selected cases. This is the latest data from the Unicure study, just showing long-run activity levels at a slightly higher level with this particular expression cassette. The approach that the company I mentioned in my disclosures, Freeline, which we set up with Wellcome Trust funding as an academic spin-out company, was to engineer an AAV vector with a higher tropism for human hepatocytes, as shown here in an FRG mouse study, also including the Padua gain-of-function mutant and a potent liver-specific promoter. Taken together, these, we estimate, enhance the specific efficiency by about tenfold. We're running a trial, a phase 1/2 trial with an adaptive design in which we've been able to vary dose level sequentially over the different patients as we target. Optimized, our intention is to target a range that is well normal. In other words, if you can attain that sort of level, you have no risk of unusual abnormal bleeding. We're using prophylactic immunosuppression for the transaminitis, which, as you've seen, can lead to loss of transgene efficiency, which is very much based on organ transplant rejection regime, but is only needed to be given for a number of weeks, not lifelong. The first cohort attained levels very similar to Unicure's levels, those two subjects, and I can report that they're both very active young men who go skiing, use the gym, and have other activities as you would expect for young active males, and have had no bleeding or use of factor IX in the approximately year and a half since they were treated. That we did use prophylactic corticosteroids, but they didn't get any elevation of ALT. And patients will do sort of hemostatic experiments in the wild, which we wouldn't do, in other words, walking into doors or occasionally getting into fights, that kind of thing. But they have not actually needed any treatment either of them. So we proceeded to elevate the dose level, and the next dose up did give us some enhancement. Now, at this dose level, 1.5 times 10 to the 12 vector genomes per kilogram, we have got a modest enhancement at dose level 2. However, it's been more than modest in one of the subjects. It's gone above the normal range, and I'm only showing you sort of point determinations here. This particular subject had a level that went considerably above that, to the point where he did need temporary anticoagulant. So you can overshoot. Both of them are free of needing any anti-treatment with factor IX. We have seen partial loss of expression in one of them during a period when we didn't have full control of the transaminitis. That lines up the first cohort with the second cohort, and you can see how fast the levels of factor IX can rise. Now, the dose level 3, just winding it down a little bit, showed a considerable drop in the response, so that we do get a kind of impression that there is a sort of there's a kind of inflection point in the response, and so we moved on from this to another dose level with another transaminitis treatment strategy. We used a prophylaxis starting at week 3. Early data suggests that this dose level is getting us right where we want to be in this sort of sweet spot. Very briefly now, as time runs out, something about factor VIII. I said it's too big to fit into AAV, so fortunately you can take out a large section of factor VIII and still have an active cofactor, which has been done for the peripheral blood replacement therapy strategies. The study that's the most advanced in this whole area has been run by our colleagues at Biomarin using an expression cassette that we designed. At 1 year, patients have achieved factor VIII levels in the normal range. Some of them have had a modest rise in ALT, which has responded to oral steroid. Annualized bleeding rates had fallen to near 0. 2-year data at this dose shows a modest drift downwards in levels, but still at this point within the normal range. A lower dose was also tried, which did show a lower response, but clinically effective. There is just most recently 3-year data now, which shows a further modest fall, but still well within a therapeutic range on this dose. We're running a study in parallel, but with a gain of efficiency modification, which is we call the V3. It's a linker which replaces the B domain and has shown to be more efficacious in terms of protein expression. We're now getting steady state levels in the low normal range at the dose shown here. We're edging the dose up. We're also working with controlling the transaminitis. Other studies that are currently concluded or ongoing are briefly summarized here, showing, as I mentioned, a great upsurge in interest and work in this area. There is one study that involves the attempt to use a zinc finger nuclease to insert an expression vector for factor 9 into the first intron of albumin. The initial result on that was not good. What about the durability? We have the longest running durability in our study, original study. Here we have data up to roughly 2 years. The Biomarin study, 3 years. That study is under consideration by regulatory authorities for market authorization, a result of which is expected in August. What are the known unknowns? Could there be consequences to integrating DNA at the chromosomes? Although AAV mostly doesn't target DNA, there's evidence that at the kind of the dose levels we're using, there is some integration. What about managing this hepatocyte toxicity? We're getting there with the immunosuppression regime. You have two alternatives really: to stay below the radar and have a lower dose, have a lower expression level, or go higher and have to have more effective immune suppression, albeit for a limited period. How are we going to overcome the issue of patients who've already got antibodies? We're looking into that, various approaches. How long will it last? Time alone will tell. How about retreatment? What about treating children whose livers are growing? And, of course, the bottom line, what will it cost? And I have a special thank you for the patients and their families, without whom none of this could have been done. My colleagues at the Royal Free UCL and at Freeline. Thank you.